DNA replication is the biological process of producing two identical replicas of DNA from one original DNA molecule. DNA replication occurs in all living organisms, acting as the basis for biological inheritance. Each time a cell divides, the two resulting daughter cells must contain exactly the same genetic information or DNA as the parent cell. For this to be achieved, each strand of existing DNA acts as a template for replication. Usually, replication occurs in three major steps: the opening of the double helix and the separation of the DNA strands, the priming of the template strand, and assembly of the new DNA segment. On the other hand, transcription is the first step of gene expression, in which a particular segment of DNA is copied into RNA by the enzyme RNA polymerase. Both DNA and RNA are nucleic acids which use base pairs of nucleotides as a complementary language. And now we're going to elaborate more on the difference between replication and transcription. DNA replication produces two exact replicas of the original double-stranded DNA molecule, where each daughter strand contains half of the original DNA double helix. Transcription produces a single-stranded RNA molecule using the double-stranded DNA. DNA replication serves to conserve the genome for the next generation of cells and individuals. On the other hand, transcription serves to form DNA copies of individual genes for immediate use in protein synthesis. During DNA replication, the products are two daughter strands that remain within the nucleus. Here, products are not degraded, while during transcription, greater part of the product, which are mRNA, tRNA, rRNA, and non-coding RNA, pass from the nucleus into the cytoplasm. Here, products are degraded after their function is over. In replication, the newly replicated DNA strand remains hydrogen bonded to its template DNA strand, while in transcription, the newly transcribed RNA strand separates from its DNA template strand. In DNA replication, the two strands are separated, and then each strand's complementary DNA sequence is recreated by an enzyme called DNA polymerase. On the other hand, in transcription, the codons of a gene are copied into messenger RNA by RNA polymerase. This RNA copy is then decoded by a ribosome that treats the RNA sequence by base pairing the messenger RNA to transfer RNA, which carries amino acids. DNA replication occurs in the S phase of the cell cycle, when the cell is preparing for division, while transcription occurs in the G1 and G2 phases of cell cycle, when the cell needs to synthesize proteins. During DNA replication, deoxyribonucleoside triphosphate serve as raw materials, while during transcription, ribonucleoside triphosphate serve as raw materials. In DNA replication, enzyme DNA polymerase requires an RNA primer for commencement of the replication. On the other hand, in RNA transcription, enzyme RNA polymerase requires no primers. DNA replication is catalyzed by enzymes DNA polymerase and DNA helicase, while transcription is catalyzed by enzymes transcriptase and RNA polymerase. DNA replication involves copying of the entire genome, while transcription involves copying of certain individual genes only. Replication involves unwinding and splitting of the entire DNA molecule, while transcription involves unwinding and splitting of only those genes which are to be transcribed. In DNA replication, the newly synthesized DNA are perfectly normal and therefore undergoes no processing, while transcription produces primary RNA transcript molecules which needs processing to acquire final form and size.